recording, so.
Thank you very much. And you'll be back in it.
Yep, I'll be back in.
Okay. Hi, guys. Can we have a sound check on the back row? How's the sound check back there? All right. So any questions on Chapter 9, the chapter we covered on Friday? Sort of the overview, state-level roadmap of metabolism. Today we're going to be beginning on Chapter 10, which has to do with the metabolism of carbohydrates. And so we're starting in on our first large area of metabolism. And given the moving around of stuff, I may put off the cell food nonsense or sense till afterwards, since we got delayed a little in starting with the IT fair that they have out there. So there we go. Okay. And a little bit further into this, I'll be giving you some hints on how to study metabolic pathways, because I know that's something that can cause a certain amount of concern for people. But I'd like to motivate you as to why carbohydrate metabolism is as key as it is. AJ, I think they still need some more handouts up there. Maybe if you could take a stack up to them. Glucose gets used for an extraordinary variety of things, all right? So it gets used for energy. The first pathway we're going to look at is glycolysis, which is an anaerobic way of using it. It's also one of the principal fuels, though not the only one, for oxidative phosphorylation, oxidative respiration, which is another pathway we'll be talking about fairly shortly. Because of its usefulness for energy, it's used for storage. And so glycogen synthesis and breakdown is an important part of carbohydrate metabolism. It gets used for structure. We use it a bit for structure, but plants use it a huge amount for structure when you think about cellulose, and we'll see why that makes sense. Glucose is also a precursor for a surprising number of different kinds of compounds. So it's not too surprising that it would be a precursor for ribose, since ribose is also sugar. Oh, shoot. Well, thank you. Thank you very much. Let's see if we can figure that out. There we go. Okay. Good thing I wasn't scrolling around with my pointer. So it is a precursor as well. That it's not too surprising that it's a precursor for ribose, which is a sugar. A little bit more surprising, perhaps, is that it's one of the major building blocks for fatty acids and for amino acids. And finally, we're going to see that it's an extremely useful source of reducing power. We talked about the need to reduce compounds during anabolism, and we're going to see that glucose is one of the major sources for reducing power through a pathway called the pentose phosphate pathway or the pentose phosphate shunt. So it's an extremely versatile molecule metabolically and well repays close attention. So we've looked at this overview roadmap before. We looked at it in the overview of metabolism chapter. And here we're going to be looking at the first class of molecules going down through that. So in this case, the biopolymers would be polysaccharides. The polysaccharides in us would be starch or glycogen. There's going to be a process for breaking them down. Phosphorylasis is a cool trick that we have to do that. That gives us monosaccharides, which are the monomers for this family of molecules. We'll be breaking down the monosaccharides through glycolysis. That's going to provide us with 2 and 3 carbon sugars. Under conditions where that's appropriate, we can take 2 and 3 carbon sugars and make new sugars. That's gluconeogenesis. And with that new sugar, we can make new polymers. So it's possible to go both directions. Those 2 and 3 carbon intermediates that we make are going to get fed into the citric acid cycle or get used to be intermediates for other things as well. So this is to orient you to where we're going to be covering in carbohydrate metabolism. So when you're learning metabolism, there are a number of questions to ask yourself about a new metabolic pathway as you meet it. What is its function in the cell? Why does the cell have it? What good does it do to the cell? What are the inputs? What gets fed into that pathway? What are the products of it? Usually the products will make sense in terms of the function for the cell. Does the pathway cost ATP or does it yield ATP? Not every pathway will have ATP involved, but many do, and that will be an important part of it. Are there steps in the pathway that involve oxidation or reduction? Again, not every pathway will, but many will, and those points will usually be informative. How is it regulated? Almost every pathway has points along the pathway that allow you to turn it up or turn it down in response to cellular demand. Since pathways have a function, you can usually see that regulation making sense in terms of that function. And finally, are there other pathways that resemble it? One of the reasons that we start with the pathways we start with is because there are enough other pathways like these pathways that starting here, you have some stepping stones to other pathways. And I'll try to point out as we go along whenever we're going to hit pathways that resemble other pathways. So before we can talk about carbohydrate metabolism, we do need to talk a little bit about carbohydrate structure. You'll have met this, I presume, in organic chemistry, but things fade with time. And we won't be able to understand the metabolism if we don't know the structures. The common feature of a carbohydrate is they are polyalcohols, a carbon backbone with a lot of alcohols decorating it. And then at least one ketone in there somewhere. Now, carbohydrates or sugars can have either a linear configuration or a ring configuration. And depending on which configuration you're talking about, different ways of representing it can be more or less useful. The Fisher representation, as a Fisher projection, is a way of representing sugars as linear compounds. They do spend some of their life as linear compounds, and different sugars spend a larger or smaller fraction as linear compounds. But they make it easy to compare the arrangement of the alcohols. The arrangement of the alcohols is a big part of which sugar is which. The others are how long it is and where the carbonyl is. So here's a Fisher projection of glucose, and you can see it's right, left, right, going on down that chain. However, a lot of sugars spend sometimes a large fraction of their time as a ring. They can interconvert between the linear and the ring form spontaneously, and depending on the sugar, they spend more or less time as a ring. So if we were to convert glucose as a linear molecule into a ring molecule, again, representing that presents certain numbers of challenges, and this is a convention that's been agreed on to represent that. And when this ring structure closes, the way in which it closes at this carbon can happen in either of two ways. And depending on the way it happens to close, the alcohol will either be down or up. And these are a very special kind of isomer of glucose. They are both glucose molecules, but they differ at that first carbon as to how the ring closed, and so they are called anomers. Anomers are isomers that differ at just the one carbon. And by convention, if the alcohol is hanging down, it's alpha D-glucose. If the alcohol is hanging up, it's beta D-glucose. Chemists count in Greek, so everything is alpha, beta, gamma something. We're going to meet more of them. But remember this alpha and beta because that's going to make a difference to the polymer structure. Now, the Howarth projection makes it look like sugars are a pancake, which is convenient but not true. Sugars, in fact, can take on two different conformations. They can either be bent a little bit in what's called the chair configuration, or they can be buckled up a little in what's called a boat configuration. And what's kind of neat about glucose is that when you look at glucose in the chair conformation, every alcohol is at the equator. The hydrogens are pointing up and down, and the alcohols are all pointing off to the sides. And so glucose has sort of the flattest, if you like, or the most relaxed conformation open to it. And maybe this is part of why glucose is such an important sugar. We don't know for a fact, but it is the only sugar that's all equatorial in the chain configuration. Now, when you have more than one glucose tucked together, you're starting to make a polymer. And the way in which your polymer is built is going to depend on which of those anomers you're putting together, whether you're using alpha anomers or beta anomers. And if you use alpha anomers, you're going to end up with starch or glycogen. And if you use beta anomers, you're going to end up with cellulose. So starch or glycogen can be described as a 1,4-alpha glucose, and cellulose is a 1,4-beta glucose. Okay? And it sounds like such a tiny difference, and yet it makes a huge difference to the actual possible structures that can be made. So if you look at the beta 1,4 linkages of as just a linear molecule, what you're getting is cellulose. And the alpha 1,4 linkages, those molecules are often branched, and it makes a more open structure, so that things like starch and glycogen are examples of alpha 1,4 linkages. Cellulose molecules can hydrogen bond side to side, and they can make very long, thin fibrils. Starch molecules tend to be more open. They're branched, which cellulose is not. And so starch in general is not as compact as cellulose. And then glycogen is even more highly branched than starch is typically. They're both starch and glycogen are alpha 1,4 molecules. But they differ in how many branches they have and who makes them. Plants make starch. We make glycogen. And some people even call glycogen animal starch. So we can actually see this at the level of what you can call secondary structure. So the alpha 1,4 linkages in starch give you a helical structure where there's hydrogen bonds between neighboring molecules along that helix. And because of that, it can be hydrated. And intriguingly, that core to the helix is big enough for an iodine atom to fit in the middle. And if you've ever done the iodine test for starch, that's what you're seeing, is that the iodine that sticks into the middle is in a different environment now, and it changes its color. This is an example of a starch grain granule. So the plant cells make a big packet of it. If you stir that starch into liquid and bring it up to boiling, the structure explodes. It becomes entirely hydrated, and instead of being a little bitty tiny starch granule, it's now this huge floppy thing, and that's why starch thickens gravies. So that's kind of a handy thing to be able to do with it. The other thing about starches is there are enzymes that will cut them up into small, 2 and 3 glucose units, and we call that malting, and that's the basis for making beer, because the amylase that breaks down the starch is heat stable. And there's a whole little story about how cool it was that somebody ever figured that out. By contrast, if we look at cellulose, it has the beta 1,4 linkages. It hydrogen bonds between polymers. So the alpha 1,4 of glucose, of starch and glycogen made hydrogen bonds up and down the helix, kind of like an alpha subunit, an alpha secondary structure in proteins. The beta 1,4 linkages make this kind of sheet-like structure with hydrogen bonds that go between the polymers. And so all the hydrogen bonds in a cellulose molecule are satisfied by other cellulose molecules. And the final structure is something that's extremely stable for that reason. That's why trees make themselves out of cellulose. So glucose polymers have secondary structure. They have local repeating structure. But unlike proteins, they come in lots of different sizes. You can have a short cellulose or a long cellulose. You can have a itty-bitty glycogen or a great big glycogen. So they do not have tertiary structure the way proteins do. They have secondary structure, but no tertiary structure. And I think we may skip over the rayon just because we had to miss that lecture back at the beginning, and I think that's a little more missable. So let's move to glycogenolysis. In many of these pathways, the name of the pathway will tell you what it's doing, or they'll have at least hints about what it's doing. And glycogenolysis is telling you what the pathway is aimed at. Glycogen is the name of the molecule. It's the thing that can give rise to sugar. And lysis in general means breaking it down. So glycogenolysis is the very short set of reactions that allows a cell to get glucose out of glycogen storage. But it does it in a very cute way. It uses an inorganic phosphate instead of water. So if you have a glycogen molecule and the right enzyme called glycogen phosphorylase can split the molecule with a phosphate. That makes it phosphorylasis. So phosphorylasis is splitting a bond with a phosphate, and the product of that is glucose 1-phosphate. So here's the Haworth projection, and the phosphate is here on the first carbon. Glucose 1-phosphate has a couple of possible uses, but it's more useful in the form of glucose 6-phosphate. So phosphoglucomutase is a mutase. It can move some things around that does that to phosphoglucose. In the liver, but only in the liver, there is an enzyme glucose 6-phosphatase, and a phosphatase cuts phosphates off, and it can do that and release a free glucose molecule. Only the liver does this because only the liver acts as a reservoir of sugar for the rest of the body. For the rest of the tissues in the body, you want that phosphate on there to keep it in the cell. And so most tissues in the body use glycogen for their own use. The liver uses glycogen to maintain glucose status in the body as a whole. Okay. So that gives us glucose either from the blood or from that process. So if we want to start using glucose, a very common entry point for doing that is glycolysis. And glycolysis, again, tells you it's breaking down sugars. This is a very, very old pathway. It's found in organisms that predate the appearance of oxygen in the atmosphere. So this pathway is over 3.5 billion years old. Almost every cell has glycolysis in one form or another. Glycolysis overall is a 10-step pathway, and what its job is to do is to take a 6-carbon glucose and to split it into 2 molecules of pyruvate, which is a 3-carbon molecule. And in the process of splitting it into 2 3-carbon pieces, it's going to produce 2 ATP. It's also going to reduce 2 molecules of NAD to NADH. And we can think about glycolysis in 2 parts. We can think about one phase where we're investing energy, or investing ATP, and the second part of it in which we get the energy back out. In addition to providing a bit of ATP, glycolysis can also provide intermediates for a lot of other pathways. I'm going to go through these reactions kind of briefly to start off with, to get you introduced to it. And maybe I'll take a moment right now to say glycolysis is going to be part of gluconeogenesis. It's going to be part of pentose phosphate pathway. It's a very key pathway. You might want to start learning it before Wednesday. Okay? You do need to learn it, but you might want to start learning it before Wednesday. So the first reaction is going to be taking glucose and adding a phosphate to it. So we have to invest the ATP here with an enzyme called hexokinase. And so here's your glucose, and after the glucose, after the hexokinase is acted, we have a 6-phosphate here, glucose 6-phosphate. Something that some people find handy for learning pathways is to play enzyme. You might want to get one of those dry erase boards and draw the first intermediate, and then play enzyme and do to it what the first enzyme does, change it on the board, and then play the second enzyme in the pathway and do what the second enzyme does, so that you recreate on the dry erase board the successive changes, because all of metabolism is done by small incremental changes along the way. Glucose 6-phosphate is not yet quite what we want to work on. We need to get fructose 6-phosphate and an enzyme called phosphoglucose isomerase. So again, it tells you what it's doing. It's taking phosphoglucose and it's making an isomer. The specific isomer is fructose 6-phosphate. And here you can see the structure of fructose 6-phosphate. It's a 5-membered ring, and this carbon's kind of flapping up in the breeze now. There's a reason for that, and we'll see why in a little bit. Fructose 6-phosphate needs a second ATP. Okay? This is a second ATP that we're going to invest during the energy investment stage. And the enzyme that does it is called phosphofructokinase. Name tells you exactly what it's doing. It's a kinase. It adds a phosphate, and it's doing it to phosphofructose. So now we have fructose 1,6-bisphosphate. And if you look at fructose 1,6-bisphosphate, you can see it's a symmetrical molecule with phosphates up flapping in the breeze on both sides and otherwise pretty symmetrical. The next step in this top half, so to speak, of the pathway is aldolase, which is actually named for the backwards reaction, which can split fructose 1,6-bisphosphate into 2 3-carbon pieces with their own phosphate on each one. So when you split fructose 1,6-bisphosphate, you get a glyceraldehyde 3-phosphate and a dihydroxyacetone phosphate. So this is an aldehyde here. So this is glyceraldehyde phosphate. And this is dihydroxyacetone phosphate. You actually want to have it all be glyceraldehyde 3-phosphate, and the last enzyme in this sort of top half of the pathway is triose phosphate isomerase. Both of those molecules are trioses, 3-carbon sugars with a phosphate on them, so they're triose phosphates. So triose phosphate isomerase is an enzyme that can turn either of those into the other one. Something that's interesting about triose phosphate isomerase is it's a perfect enzyme. It's basically limited by the access of diffusion. It's so good at doing what it does. So this is the first phase, the energy investment reactions. Adding the ATP does a couple of things. One, it keeps the glucose inside the cell. Glucose by itself in many cells could go right back outside the cell through the glucose transporter if the concentration got higher than outside. The phosphate prevents that. The second thing is that phosphates that are added set up the molecule for the subsequent reactions. So these reactions we're going to look at now are the payoff steps. This is where you actually get something out of glycolysis. And some of them are rather cool reactions indeed. So we have these 2 molecules now of glyceraldehyde 3-phosphate. Remember, we turned the DHAP into glyceraldehyde 3-phosphate. And a lot has to happen at once. This is perhaps the busiest arrow you'll see in metabolism. We're going to be using inorganic phosphate and an NAD+, and we're getting out an NADH, so something got oxidized because this is now reduced. This is the oxidized form. This is the reduced form. Somewhere on that molecule we had some reduction go on. And the phosphate gets taken up, so we end up with molecules of 1,3-bisphosphoglycerate. And here's what that looks like. It's a 1,3-bisphosphoglycerate. This end of the molecule has been set up in such a way that it's a high-energy bond. It's equivalent in energy, or pretty close to equivalent in energy, to the energy of an ATP. And so we can make ATP at this step. If we feed in 2 molecules of ADP, 1 for each of the 3-carbon pieces, we can get out 2 ATP. We finally have broken even. We invested 2 ATP a couple of steps. Now we're getting them back out. This enzyme is called phosphoglycerate kinase. It's actually named for the backwards direction here. So now we have 2 molecules of 3-phosphoglycerate. So it's glycerate because it's a carboxylic acid, and the phosphate's on the 3-carbon. 3-phosphoglycerate can be rearranged into 2-phosphoglycerate. That's just a mutase, a reversible reaction. So now we have 2 molecules of 2-phosphoglycerate. And we're going to do kind of a cute trick. We're going to take a water molecule out of it through an enzyme called enolase, and that will rearrange it into 2-phosphoenolpyruvate. So here you see phosphoenol pyruvate. The enol is the tautomer with a keto group. And in this case, we've locked it into the enol state because there's a phosphate on the alcohol. And so 2-phosphoenolpyruvate is energetic enough that we can get another ATP made. So each molecule of phosphoenolpyruvate makes an ATP. And the product of that reaction is pyruvate. Again, an enzyme named for the backwards reaction, as if you're starting from pyruvate and going to phosphoenolpyruvate. So those are the 10 basic reactions. We've got the 2's in front of these things because these are all the 3-carbon backbones. We started with 1 6-carbon glucose, so that's to help you keep the bookkeeping straight. But we're going to look at these each in a little more detail. So I'm going to try to look at each of the reactions and sort of flesh them out for you. This is hexokinase. So glucose with an ATP is going to be phosphorylated to glucose 6-phosphate. The molecule hexokinase actually closes like a clamshell around the substrates in order to do that. And the free energy change, in other words, the energy difference in the cell, is a minus 17 kilojoules per mole. So this is a very favored reaction, very strongly favored reactions. And in metabolic terms, it's essentially irreversible. That's why we draw it with a one-way arrow. So the phosphate allows to recognize the substrate, and it also is going to change the reactivity of the compound as we go along. It's important to note that if you get your glucose 6-phosphate from glycogen, you did not have to pay ATP. You used an inorganic phosphate in phosphorylasis, so a glucose that comes from glycogen actually gives a little higher energy yield than a glucose that comes in from the bloodstream. You had to pay the energy up front when you made the glycogen, but you don't have to pay it when you need it for glycolysis. So phosphoglucose isomerase is rearranging an aldose into a ketose from a glucose to a fructose. Free energy associated with it, nearly zero, close enough. This is freely reversible under the conditions of the cell. And it's a little easier to see what it did if you look at the Fisher projection instead of at the Haworth, because you can see here that in the glucose, this is an aldose, meaning that's an aldehyde. It's on the end of the molecule. And fructose is a ketose, where the carbonyl is internal, so it's a ketone. Phosphofructokinase. This is a very important enzyme. Phosphofructokinase tells you what it does. Kinase adds a phosphate to phosphofructose. So it's going to take fructose 6-phosphate and add a phosphate to make fructose 1,6-bisphosphate, and then an ADP left over. Again, like our first kinase, like hexokinase, a very large negative delta G. Again, this step is considered metabolically irreversible. This is a very highly regulated enzyme. We sometimes call it the committed step for glycolysis. You might wonder, why wasn't hexokinase the committed step? We're going to see there are other things you can do with glucose 6-phosphate. So although glucose 6-phosphate is part of glycolysis, it's also part of some other things as well. Phosphofructokinase is the committed step for glycolysis as a pathway overall. Some of this regulation makes sense in terms of the kinds of things that phosphofructokinase is used for. ADP is a product of the enzyme, and yet it increases the binding of fructose 6-phosphate. So that's kind of odd, right? It's product stimulation instead of product inhibition. But that's because ADP is a sign that energy is needed by the cell. And for it to increase the binding of fructose 6-phosphate, it binds at a different site. It's an allosteric regulator of phosphofructokinase, in addition to being a product of it. Phosphoenolpyruvate is an end product of glycolysis, and it will decrease fructose 6-phosphate binding. Abundant levels of phosphoenolpyruvate would be a sign that there's a lot of energy available, or the end product is available. We're going to see that in mammals like us, there's an additional kind of regulation in a very sneaky way. That fructose 2,6-bisphosphate is is another level of regulation on top of these simple allosteric ones that are through metabolites. And this is just a summary of some of these different regulators. That ADP or GDP-A and P are all signs of low energy. Fructose 2,6-phosphate is an allosteric regulator that is not a metabolite in the usual sense. That's regulated by, for instance, the liver and by hormones in the blood. We've mentioned phosphoenolpyruvate. ATP is a sign that energy is abundant, so it can decrease activity. Some other things that can regulate it are citrate and fatty acids. Those are both fuels of other kinds that would be reasonable signals for phosphofructokinase. or PFK. This is a drawing of phosphofructokinase, and you can see that in addition to catalytic sites where the substrates can bind, there are a number of allosteric sites that are not active sites. They're allo sites. They're other sites at which regulators can bind. This is one example of the kinds of change in kinetics that under low concentrations of ATP, this would be the enzyme activity with changing concentrations of fructose 6-phosphate. And so you have the typical hyperbolic kinetics of a simple reaction. And as you add ATP, it becomes more sigmoidal in its kinetics. Its Km is affected, but not its Vmax. So you can overcome this effect of ATP with AMP, which is a low-energy form of adenosine nucleotide. And so the ratio of those two is a way of kind of like a thermostat for the cell energy, for the cell to know how much energy it's got and therefore how active glycolysis should be at any given moment. AMP is a good signal because there's a way you can convert ADP into ATP if you have several molecules of it around. So ADP has one high energy bond, and adenylate kinase can rearrange those to give you AMP and then ATP. So this is one reason why AMP is a particularly strong signal for low energy status. This is showing you this pathway schematically to emphasize some more of the regulation. So you've got fructose 6-phosphate going to fructose 1,6-bisphosphate, and a very peculiar compound. Here we go. This is fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is made by a separate enzyme called phosphofructokinase 2 to emphasize that it's going to add the phosphate to the 2 position instead of the 1 position. And the activity of this enzyme is regulated at a higher level through things like hormones and stuff like that. And fructose 2,6-bisphosphate is a positive regulator of phosphofructokinase 1. So fructose 2,6-bisphosphate will increase the amount of activity for that enzyme. And we've seen these graphs before, but I think this kind of draws together in one set of graphs several of these regulators at once. So you can see here, here we're seeing the effect of fructose 2,6-bisphosphate. This is just an allosteric regulator. It's not a product. It's just an allosteric regulator. In the absence of it, it has a fairly high Km or a fairly low affinity. And with even small amounts of it present, very big increases in affinity, and therefore the concentrations of the cell, probably activity as well. Here's the one that helps you see the relationship between fructose 2,6-bisphosphate and ATP. So in this case, we were increasing the concentrations of fructose 6-phosphate, which is the substrate for it. Here, ATP is another substrate for it, but it's also an allosteric regulator. And we see that because after an initial increase in enzyme activity, it actually comes down, which is pretty peculiar for a substrate. So in this part of the curve, we're seeing the allosteric effect overwhelming the fact that it's a substrate. And what the fructose 2,6-bisphosphate, which is just an allosteric regulator, can do is greatly relieve that inhibition. So you can see that as we increase the amount of fructose 2,6-bisphosphate, that the activity curves rise quite a bit higher. Okay. This is just showing you the special enzyme that's used to make fructose 2,6-bisphosphate. It's kind of a funny enzyme because it's both the synthetic and the degradative enzyme at the same time. It has one domain that makes fructose 2,6-bisphosphate and another domain that breaks down fructose 2,6-bisphosphate and a regulatory region that allows the cell to control which of those two activities is actually going on at one time. A very busy little molecule, kind of like a setback thermostat. So now we have our fructose 1,6-bisphosphate. We have a primed 6-carbon sugar. We're going to break fructose 2,6-bisphosphate apart into 2 3-carbon fragments through this enzyme called aldolase. The activity of aldolase is the reverse of an aldol condensation, of an aldehyde and alcohol being condensed. So it's named as if we're going in this direction. During glycolysis, we're going in that direction. Aldolase has a standard free energy change of 23 kilojoules per mole. That is not a very favorable reaction, but that's the standard free energy change. The actual free energy change for aldolase in a cell that's actually doing glycolysis is actually less than 0, and that's because its products are withdrawn so rapidly that it pulls the reaction in the forward direction. So this is one of the more striking examples of where the actual free energy change is very much affected by the conditions in the cell. This is a dramatic example of why the standard free energy change and the actual free energy change can be very different. And in the cell, what matters is the actual free energy change. Because we lost that day back at the beginning, I'm probably not going to go through quite the same level of detail with the enzyme mechanisms as I might have if we had a full 3 days for carbohydrates, which is how I usually teach this. So what I'm going to try to do is highlight some of the sort of key features of the mechanism rather than walk you through the whole mechanism. Aldolase is another example of an acid-base and covalent catalysis, like we saw with chymotrypsin. And the fructose 1,6-bisphosphate can bind in the linear form, and we're going to see that lysine and tyrosine are both important to the active site. You remember you have the catalytic residues that actually catalyze things, and then you have a substrate binding that can influence who actually gets to sit in there. Well, some of the catalytic residues in aldolase are a lysine and a tyrosine. And the lysine is going to end up becoming a covalently attached to the carbon chain. And the tyrosine is actually going to be acting as an acid base. So here you can see the lysine has been covalently attached to the fructose. And here you see the tyrosine pulling off a proton, so it's acting like a base in that point. In doing that, this 3-carbon piece gets broken off from the backbone. And so we're going to release the first product, the aldehyde. The other 3-carbon fragment is still attached to the lysine. The tyrosine gives back the proton. And we can then hydrolyze that base by adding in a water molecule. And the second 3-carbon fragment, dihydroxyacetone, comes off. So the lysine and the tyrosine cooperate, with the lysine being the site of covalent attachment and the tyrosine acting as acid base. So now we have one molecule of dihydroxyacetone phosphate and one molecule of glyceraldehyde 3-phosphate, but what we want is glyceraldehyde 3-phosphate. There are other things we can do, and we'll see them later on with dihydroxyacetone phosphate, but those are other things. For glycolysis, we need to get it into glyceraldehyde 3-phosphate. To do that, we need this enzyme triose phosphate isomerase. Both of them are trioses, 3-carbon sugars, both phosphates, but they're isomers, and that's what this enzyme is going to do. It's an incredibly beautiful enzyme. It is only limited by how fast the substrate gets to it. Extraordinarily good enzyme. The free energy change here is about 4 kilojoules, which isn't great. But the glyceraldehyde 3-phosphate is going to get consumed rapidly, and so that pulls the reaction through. Okay. That was the energy investment stage when we're setting things up so we can actually get something out of it. All we've done so far is spend money. At this stage, we're going to start doing things that are going to allow us to get something out of the glucose, or what you could call the payoff stage. The next enzyme we're going to talk about is glyceraldehyde 3-phosphate dehydrogenase. So again, the enzyme tells you what it's going to do. Glyceraldehyde 3-phosphate is its substrate, and it's a dehydrogenase. It's going to rip some hydrogens off of the substrate and put them onto some kind of an electron carrier. Whenever you see dehydrogenase, you can be sure that someone somewhere is getting oxidized or reduced. So this is going to be an oxidation reduction reaction. Everything from this point on are going to be trioses, or 3-carbon compounds. We're going to end up making a total of 4 ATP. We invested 2, so that'd be a net of 2 ATP. If we got our sugar from glycogen, it would be a net of 3 ATP, right? This second part of it, the second set of reactions, is the most conserved when you compare across organisms. So there's some thought that this may be the most ancient part of the pathway, that the top part of the pathway evolved more recently. So what's going to happen? Glyceraldehyde 3-phosphate dehydrogenase does a very cute, strange thing. Here we have an aldehyde. This is glyceraldehyde 3-phosphate. We have an NAD plus and an inorganic phosphate. And this enzyme is going to mush all that stuff together to give us 1,3-bisphosphoglycerate. So it's added a phosphate to this aldehyde, and it's reduced the NAD plus to NADH. And if you look at this carbon, you can see that that's the carbon that got oxidized. See, now that carbon has a bond to an oxygen instead of to a hydrogen. So this is the carbon that got oxidized, and the oxidation of that carbon is what provided the energy for the NADH. And the source of that phosphate is just inorganic phosphate. So there's a couple of dramatic things that inorganic phosphate does. Phosphorylasis when we break down glycogen, and then being the source for glyceraldehyde 3-phosphate dehydrogenase. ATP gets a lot of glamour and glory, but inorganic phosphate can do a few interesting things. So glyceraldehyde 3-phosphate is going to have both oxidation and phosphorylation happening to it. The glyceraldehyde 3-phosphate is going to bind to the active site, which already has an NAD plus sitting there. And there's a cysteine in there that's going to make a covalent bond to the carbon where everything exciting is going to happen. So this has an active site cysteine. And the product of that is going to be an acyl phosphate. And an acyl phosphate like this has a high energy bond. So the carbonyl oxygen, oxygen phosphate, that's a high energy bond. Set up like this, the NAD plus can take out some electrons, and in doing that, it's created a thioester intermediate. Now, you remember when we talked about the overview of metabolism that thioesters, for some reason, we don't fully understand, are high-energy compounds pretty equivalent in energy to ATP. So here we have inside the enzyme as a covalent attachment a thioester because the NAD plus picked up the electrons and hydrogen and left it in this form. This thioester intermediate is part of the reason the enzyme can get away with this. If it tried to go to oxidize it and then straight to the acyl phosphate, this would have been a very large delta G in between. But the thioester intermediate makes the individual steps of this much more doable. NAD and NADH are both water soluble, so that reduced NAD, the NADH, leaves. A fresh molecule of NAD plus comes in, and it's going to then allow the phosphate, it's going to sit there in the active site. Inorganic phosphate comes in to attack that carbon that's in the thioester. The thioester has that high energy thing, so we can now have 1,3-bisphosphoglycerate form and leave, and the active site is regenerated for future use. So, very cute trick. Very cute trick. Keep in mind we made an NADH. We can't do anything with that NADH right away, but it will be important in a lot of different ways. But now we have 1,3-bisphosphoglycerate. And 1,3-bisphosphoglycerate at this end of the molecule has a high energy phosphate bond. Because that high energy phosphate bond is there, we can add an ADP and make an ATP and a plain old 3-phosphoglycerate. The enzyme is named backwards. It's named as if you're starting from 3-phosphoglycerate and making 1,3-bisphosphoglycerate, so just be aware of that. For glycolysis, it's named backwards. If you look at this end of the molecule, this is a glycerate. That's a carboxylic acid. It was a glyceraldehyde. So you can see that was an oxidized carbon. It's the oxidation of that carbon that provided the energy to make that ATP. This is an example of substrate-level phosphorylation. Substrate-level phosphorylation is when you make a molecule of ATP from another substrate. We're going to see that it's different from oxidative phosphorylation or photophosphorylation, which are the main energy sources. But substrate-level phosphorylation allows you to make an ATP or equivalent high-energy bond from another substrate. The free energy change for this is minus 19 kilojoules, so it really likes doing this. And this helps to pull the glyceraldehyde 3-phosphate dehydrogenase forward. GAPDH is not that is a somewhat disfavored reaction, but the phosphoglycerate kinase has such a negative delta G that it helps pull it forward. So these can be considered coupled reactions. The next one is a very simple little reaction. We're going to move a phosphate from the 3 to the 2 position. That's a mutase, phosphoglycerate mutase. So we take 3-phosphoglycerate, make a 2-phosphoglycerate. These are equivalent in their energies. This is a fully reversible intermediate, fully reversible reaction. And it takes advantage of a histidine. So 3-phosphoglycerate can pick up a phosphate from a phosphohistidine and park the extra phosphate on it to make so that you can bop back and forth between the 3 position and the 2 position. They're equivalent. So here's another interesting role for histidine, acting as a temporary parking place for a phosphate. So now we have 2-phosphoglycerate. And we're going to go from 2-phosphoglycerate to phosphoenol pyruvate. This is an enol, right? A double bond and an alcohol with a phosphate on it. So phosphoenol pyruvate. And enolase is the enzyme that can do this. Interestingly, what enolase does is it takes out a water molecule between these 2 carbons, and that sets up this double bond to the phosphate. This is a freely reversible reaction. You can think of this as kind of an internal oxidation reduction reaction, maybe, is another way of thinking about it. But what we've done is to set up another high-energy phosphate bond. This phosphate would really like to leave. So now we have phosphoenol pyruvate. And if we add another ADP, pyruvate kinase can make ATP out of it and pyruvate. Again, it's named for the backwards reaction. In glycolysis, it's going to go from phosphoenol pyruvate to pyruvate. This is our second example of substrate-level phosphorylation. And the reason it goes forward is that after that phosphate is transferred to ADP to make ATP, you have enol pyruvate, and enol pyruvate is not a very stable form. It's going to tautomerize into pyruvate very rapidly. So it's going to pull off the product very rapidly. And this tautomerization helps pull the reaction through overall. So this is just giving you a visual cue to the stages. These are the actual shapes of the enzymes that are used in glycolysis. You might want to use these as a review for it. I notice that we're getting on to 4:20, so maybe it's time to, I'll let you take a look at those on your own time. I guess I'll see you Wednesday.